Hello everyone, welcome you all. So today we are going to solve this question which has appeared in GATE examination in 2014 in the Excel question paper. This question which I have taken is question number 20 from the biochemistry section. Okay, so let's read it out. So this question states that a 7 kb DNA molecule of a specific sequence has 2 eco-R1 and 1 PVU restriction endonuclease sites. So restriction endonuclease sites are given in a 7 kb DNA molecule. Okay. So the restriction sites are shown below. So these are the restriction sites which they have given. So the DNA was completely digested with both eco-R1 and PVU2. The digestion product was purified and added to a appropriately buffered reaction mixture at 37 degree, which contained the clean off fragment of DNA polymerase 1 and alpha 32 phosphorus DNTPs. After 1 hour, the DNA in the reaction product was purified and analyzed by electrophoresis. So the bands were visualized by both ethidium bromide and ethidium bromide staining and autoradiography. The results are shown below. So these are the images of the result. And it has been asked to us about the correct map of the DNA molecule. So these 4 options are given to us. Okay. So as this question is talking about the restriction endonuclease digestion. So let's first talk about restriction enzymes. Restriction sites, restriction enzymes, and specifically these are restriction endonuclease enzyme. Endonuclease means that is making a cut inside the DNA. Okay, like here it is internally cutting the DNA. Okay, and exonuclease on the other hand, it like cuts the DNA from the outside. Okay, so like making it a little shorter. So this is the exonuclease and endonuclease. So restriction enzymes are generally endonuclease enzymes. So restriction sites are the specific DNA sequences which are generally 6 to 8 base pair long, which can be identified by restriction enzymes, and restriction enzymes will make a cut on those specific restriction sites. Okay, so this, suppose I'm making a double stranded DNA which has a restriction I'm trying to show you a double stranded DNA here which have this sequence. So this is a specific site which can be recognized by BAMH1 enzyme, BAMH1 restriction enzyme. Okay, and BAMH1 can recognize the site in double stranded DNA and make a cut at these 2 places. Okay, and now after this site gets recognized by BAMH1, it will get digested, or the DNA will get digested, and it will produce the ends like Okay, so these kind of digested fragmented DNA gets produced after the digestion. Okay, so these restriction enzymes comes under the category of type 2 restriction enzymes. So in type 1, what we have, type 1 makes a cut away from the restriction sites, away from recognition site, but type 2 restriction enzymes make cut um, in the recognition site. Okay, so BAMH1, EcoR1, all these are the type 2 restriction endonuclease which recognizes a specific sequence and make a cut at those specific sequence only in the DNA. Okay, so this is the BAMH1 specific restriction site. So likewise, all other restriction enzymes have their specific unique restriction site that they can recognize and make a cut in the DNA. Okay, so in this question, now we got the point of restriction endonuclease sites which are given here. So this is the EcoR1 restriction site is given and PUV2 restriction site is given. Now with the arrows you can see where the enzyme will gonna cut it. Okay, so as I have shown one example of BAMH1 restriction digestion, similarly this is the EcoR1 restriction digestion sites. Okay, so in the BAMH1 what happened here, that something this kind of cut got created. Okay, so this we call as sticky ends. Okay, sticky ends of the DNA. This is a single stranded DNA. Half of the portion is a single stranded DNA. Okay, so these are called as sticky ends, but as you can see here in the PUV2, so here what is happening after the restriction digestion, it will produce a sequence like this. Okay, it will just get divided in equal half without producing any overhang. So this is called as blunt end. Okay, and while EcoR1 after the digestion it will produce an end like this. Okay, and this. So this is called a sticky end. So these are also two different kind of ends that produce after the restriction digestion, blunt end and the sticky end, which you can see here. Okay, so now let's talk about the so they have digested. Okay, so they have certain DNA molecule, a certain size, they have digested with these two enzymes, and now after the digested product they have put it under such condition where they have provided the reaction the clean off fragment of DNA polymerase and alpha 32p labeled DNTPs. Okay, so these DNTPs are the radio labeled DNTPs. Okay, so that means wherever these DNTPs will get associated, so those fragments will become radio labeled. So in whichever DNA fragment these DNTPs will associated, it will make them radio labeled DNA. Okay, so what is Greenhoff fragment of DNA polymerases? Okay, so Greenhoff fragment of DNA polymerase. Okay, so let's first talk about DNA polymerase. DNA polymerase 1. So DNA polymerase, we know it, polymerase means polymerize the DNA strand. Okay, so DNA polymerase 1, it has certain functions. So it has a DNA dependent DNA polymerase activity. Okay, so it needs a single standard DNA template, and that using that template it will polymerize the next strand or the complementary strand via the polymerase activity, and this activity it has in a 5 prime to 3 prime direction. Okay, first point. Second point, this DNA polymerase has one exonuclease activity, or we can say proofreading activity. Activity which is in 3 prime to 5 prime direction. Okay, so this mediates the proofreading. That means while adding the complementary bases to the DNA, if there is any mismatch via the proofreading activity, that mismatch will be removed and removed via the exonuclease activity, and another strand of or the correct base of the DNA will be added via the polymerase activity. So this is the exonuclease. So at the end of the DNA, if there is a mismatch from the end of the DNA polymerase 1 will perform its exonuclease, remove that mismatch pair, and again perform the polymerase activity. So this exonuclease of proofreading activity is the 3 prime to 5 prime direction. It also has another exonuclease activity in uh, 5 prime to 3 prime direction. Okay, so this exonuclease activity is for the removal of the RNA primers from where the uh, from where the DNA polymerase sits and starts it performing its activity, because we get one RNA polymerase as a RNA sequence as a primer, which helps or which provides a 3 prime end to start the DNA synthesis on the DNA. Okay, so now when the DNA replication process or DNA polymerization process gets complete, that hybrid fragment or the RNA primer needs to be removed so that the DNA sequence can be synthesized. Instead of RNA, DNA, it becomes the DNA sequence. So that the RNA primer removal is done by the 5 prime to 3 prime exonuclease of the DNA polymerase 1. So these are the DNA polymerase. Now, what clean off fragment? Clean off fragment is the largest fragment. Okay, so it is the largest fragment of DNA polymerase 1. Okay, so this clean off fragment, it has this suppose if I have this DNA polymerase, okay, and this section contains the polymerase activity. This section contains the exonuclease activity of 3 prime to 5 prime. And this contains the exonuclease activity of 5 prime to 3 prime. Okay. So this entire thing is DNA pol 1. Then this much portion is clean off fragment. Okay, so now our clean off fragment harbors the DNA dependent DNA polymerase activity, and it also harbors the exonuclease or the proofreading activity. Okay, but it does not possess the 5 prime to 3 prime exonuclease activity which is present in DNA polymerase 1. So this is the clean off fragment. So clean off fragments, the basically what it will do, it will make the sticky ends of your DNA blunt, and it is generally used for the probing of the DNA, to make the DNA probes. Okay, so suppose I have two kinds of DNA. So Okay, so these are two different overhangs which is present in DNA. So this one where the 5 prime end is the single stranded, this is called 5 prime overhang. Okay, so this is called 5 prime overhang, and the other one which is called as 3 prime overhang. So these so clinofragment will gonna perform its activity. So as you can see here, it has the DNA dependent DNA polymerase activity. So that activity can be performed in 5 prime to 3 prime direction. So if we'll see these two figures, in these two figures, so uh, first if we'll talk about this exonuclease activity, so exonuclease activity which is possessed in 3 prime to 5 prime direction. Okay, that means this 3 prime to 5 prime direction, in this direction. Okay, so here in this direction it can perform its exonuclease activity. So it will chop off this 3 prime overhangs, make it blunt, something like this. Okay, and if on this sequence if we'll see 3 prime to 5 prime exonuclease activity cannot be performed, but uh, 5 prime to 3 prime, but 5 prime to 3 prime polymerase activity can be performed. Okay, so it will add the new bases here in the DNA. Okay, and it will make the double stranded DNA, and then also it will produce those blunt ends. Okay, if you see, the main function is producing the blunt end. So if 3 prime overhang is present, it will chop it off via the exonuclease activity and give a blunt end DNA like this. And if it is 5 prime overhang is present, the exonuclease activity will not work, but the polymerase activity can be performed, and by polymerizing it will make the double stranded blunt end DNA. So that can be done by the DNA clinofragment of the DNA polymerase. Okay, so if in our question, if we'll see, so in our question, so this DNA molecule is subjected to digestion. Okay, so 7 kb DNA molecule, and after the digestion, after the clinofragment provided and the DNTP provided, what will happen? And the DNTP provided, so this eco-armor PUV2 ends will be produced. So as these PVU2 ends are already blunt ends, clinofragment will have no activity. Clinofragment uh, will not perform any work on this particular generated fragment, but on this one, okay, the clinofragment will perform the polymerase activity, because these are 5 prime. So, so here it will perform its DNA polymerase activity in 5 prime to 3 prime direction. Here also 5 prime to 3 prime direction, and make the double stranded DNA. Make it blunt end. Okay, so that is what going to happen. So now after this particular thing, they are running the fragments on the ETBR gel. So let's first see the ETBR gel. So in ETBR gel we are able to see 4 fragments. So how ETBR stain gel DNA will visualize? So ETBR intercalates with the DNA molecules, and those intercalated DNA molecules with ETBR, when we visualize under UV, we are able to see the DNA bands. Okay, so there is nothing, radio labeling is required. All kind of DNA you can see because everything will be intercalated via ETBR. Okay, so there are 4 fragments that are being produced. Okay, 4 fragments. It has been given to us 7 kb DNA molecules, and we have 2 eco-R1 and 1 PUV site. 2 eco-R1 and 1 PVU, 2 sites. That means total 3 restriction sites. Okay, and the fragments which is produced is 4 fragments. If it is a circular DNA, it would have produced the similar number of fragments as many as the restriction sites are present. But it is producing one more fragment. That totally indicates that it is not a circular DNA, rather it is a linear DNA. So now these 2 options are rejected, and we can see these 2 options. If we'll see the sizes, let's see if the sizes given are the correct. So here we'll have a 3 kb fragment, a 2 kb fragment, 0.5 kb fragment, and 1.5 kb fragment. Okay, 3 kb, 2 kb, 0.5 kb, 1.5 kb. What about this one? So here also we are getting 3 kb fragment, 2 kb fragment, 0.5 kb fragment, and 1.5 kb fragment. So if we'll just see the ETBR gel, any of these 2 options could be correct. eco-R1, eco-R1 and then PVV2, or eco-R1, PVV2, or eco-R1. Which one is correct? We cannot conclude seeing the ETBR staining gel, but we can definitely conclude that it is not a circular plasmid, it is a linear plasmid or linear DNA. Okay, so now the autoradiogram, it is showing us only 3 bands. Okay, so as now we have discussed, let's see. So if it is this one is getting digested, it will produce eco-R1 site sticky end here, eco-R1 sticky end here, eco-R1 sticky end here, eco-R1 sticky end here, PUV blunt end here, PUV blunt end here. Okay, while this one eco-R1, eco-R1, PUV, PUV, eco-R1 sticky end, eco-R1 sticky end. So if we'll see in this one, as this DNA strand have the eco-R1 sticky end here, so here Clino fragment will polymerize it, and the radio-labeled DNTPs will incorporate. This fragment will be visible on the autoradiogram. This one also have eco-R1 sticky end, will be polymerized and gets incorporated with radio-labeled DNTPs. This will also be visualized on autoradiogram. And these two also have eco-R1 sticky end, where Clino fragment will perform its activity and radio-labeled DNTP will be incorporated in these 2 strands. So if we'll see here, all 4 should be incorporated with the radio-labeled DNTPs, and it should be visualized on autoradiogram. But we are only seeing 3 bands. Let's see the other DNA. So here also eco-R1 sticky end is there. Clino fragment will perform its activity. Radio-labeled DNTPs will be here. Here 2 eco-R1 sticky hands. This will also become radio-labeled. This one as well become radio-labeled. But this one, which has PUV digested blunt end at the end, so here no work of Clino fragment or no activity of Clino fragment will be happen. So this will not be radio labeled. This fragment of the DNA won't be radio labeled. So this 1.5 kb fragment will not be able to see on the DNA, but all these 3 will be able to see. So this is the size of 1.5 kb band. If we'll see here, 1.5 kb, this band only missing here in the autoradio gram, but 3 kb band which is there, 2 kb band is there, and 0.5 kb band is also there, not the 1.5 kb which is supposed to be digested by PUV2. So if we'll see the we'll compare ETBR stain and autoradiogram, and this particular DNA map is correctly matching with our results which they have got after the on the TBR gel stain and the autoradiogram. So the correct option would be B, a linear DNA molecule which is having EcoR1 to EcoR1 restriction site subsequently, and then a PUV2 restriction site. So this is our correct option. I hope you understand the answer, and thank you for watching. This is my NPTEL reference. Thank you.